Coral reefs are one of our most important resources here in the Virgin Islands. They provide important habitat for countless underwater creatures, support commercial and recreational fisheries, provide protection from storms and erosion, and account for millions of dollars in tourism every year. Despite the important role corals play, they are disappearing at an alarming rate. Climate change causes coral bleaching and stronger and more frequent hurricanes. Overfishing and destructive fishing practices remove fish that play an important role in the reef ecosystem. Pollution, whether it's a plastic bag floating in the water or runoff from a construction site, can have detrimental effects on coral reefs. Coral diseases, which are still poorly understood, have spread rapidly across the region, devastating key reef-building corals. Although recent decades have painted a pretty dismal picture for coral reefs, there are ways we can help. We can reduce our carbon footprint. We can choose locally caught, sustainable seafood. We can prevent waste and pollution and support our marine parks. Once we are doing all we can to protect reefs, then we can take it a step further and actively restore coral reefs. Coral restoration has to work in conjunction with other efforts to lessen the threats to coral reefs. The Nature Conservancy started its coral restoration program in the USVI in 2009 with the first nursery-grown corals outplanted in 2012. Now, a decade later, over 30,000 corals have been outplanted onto reefs around St. Croix and St. Thomas. Other partners and agencies have joined the effort, expanding the capabilities of restoration. Now there are multiple field and lab-based coral nurseries, increasing the number and genetic diversity of corals we can use to restore reefs. When you first look at a coral reef, it can appear to be just a lot of colorful rocks. But corals are made up of thousands of individual animals called polyps. The polyps are made of soft tissue and consist of a central mouth surrounded by tentacles. To protect their soft tissue, polyps secrete calcium carbonate to create a cup-shaped hard skeleton. As polyps grow and multiply, they become a coral colony. Over time, as more and more colonies continue to grow and create more skeleton, a reef is formed. Inside the tissue of each polyp live microscopic algae called zooxanthellae. Similar to the pigment of your skin, zooxanthellae give corals their color. They also photosynthesize and create food for the coral. This symbiotic relationship means that corals are, in a way, both plant and animal. An animal with tiny plants living inside its tissue. The relationship between the coral and the zooxanthellae is mutually beneficial. But if conditions become unfavorable or if corals get stressed, the coral can expel the zooxanthellae, making the coral appear bright white, since you can see through the soft tissue to the hard skeleton. This is what's referred to as coral bleaching. As all animals do, corals need to reproduce. Corals can reproduce sexually or asexually. Sexual reproduction usually happens through mass spawning events, where coral individuals release male and female gametes into the water column, and through mixing, they fertilize and settle back onto the reef. Asexual reproduction includes budding, where polyps clone themselves to expand and grow the colony, and fragmentation, when a part of a coral colony breaks off and reattaches to the reef and continues to grow as a separate but genetically identical colony. Fragmentation can happen naturally during high wave energy events. Most restoration efforts have focused on taking advantage of asexual reproduction techniques. We collect broken fragments of coral, break or cut them up even further to create many individuals from one broken fragment. Since each of those individuals would then be a clone, it is important to collect fragments from multiple parent colonies or multiple genotypes to ensure genetic diversity in the nursery and the outplant sites. We can go out after storms, large swell events, or even boat groundings to search for these fragments of opportunity that can be multiplied many more times in the nursery. Sexual reproduction also plays an important role in coral restoration. By collecting gametes during coral spawning events, we can assist fertilization and settlement of coral recruits. Sexual reproduction is the only true way to increase genetic diversity, so this is an important part of a restoration program. Higher genetic diversity of outplanted corals will increase those corals' chances of surviving, but also increase success of natural sexual reproduction in the future. Collected corals are fragmented further using a variety of methods. Branching corals are easily fragmented using garden shears or wire cutters to clip off individual branches from a colony. Boulder corals can be fragmented underwater using a hammer and chisel, but can also be brought onto the boat or into the lab where you can use large saws to make precise cuts. Tile saws or band saws can be used to cut corals into tiny pieces down to individual polyps. Cutting boulder corals into small fragments and then planting them on existing skeletons and allowing them to grow together can reskin these large boulders, creating reproductive-sized corals in a fraction of the time it would take a coral to grow from a single polyp. Land-based coral facilities are increasing the capabilities of restoration projects. By performing fragmentation as well as sexual reproduction techniques in water tables, you can increase the production and quality of nursery-grown corals. Water quality control and being able to perform fine tasks without the use of scuba can exponentially increase nursery production. It can also allow controlled conditions for tiny corals to grow larger and healthier before being returned to the reef. Since you don't have to be on scuba, many more people can get involved with restoration efforts through the land-based nursery. Volunteers can help with monitoring water chemistry, maintaining and cleaning substrates and tanks, cutting and measuring corals, and collecting health and growth data on coral fragments. In-water field nurseries should be accessible, appropriate depth for the species of coral you're working with, and should have good water quality. The structures that hold the corals can be made of a variety of materials, but some of the most common include PVC, fiberglass, rope, and rebar. Coral trees and other floating structures are very common for growing staghorn and other branching corals. They are made from PVC, fiberglass, and monofilament lines. By hanging the corals, they have 360 degrees to grow and are up off the bottom away from sand and predators. Other nursery structures sit on the bottom, including tables and frames. For non-branching corals like boulder corals, brain corals, and other massive corals, tables can provide a more stable base for holding a coral fragment that can't be easily attached to monofilament line. The A-frames and dome structures can provide coral for outplanting without having to remove entire corals, and these structures require little to no maintenance. No matter the structure, it is essential that coral nurseries are maintained regularly to keep them free from algae, fire coral, sponges, and predators including snails and fireworms. Using brushes and scrubbies, divers clean around the coral fragments to give them more room to grow and prevent competitors like algae from taking up their space. Maintaining a nursery also includes monitoring the health and growth of the corals. We are always on the lookout for predators, disease, and bleaching. We can track the growth of corals by measuring or using photography. So, if you are ever in the nursery and you see any unhealthy corals or any structures that are in need of repair, make sure to let us know by writing down the structure number and a description of the issue. When working in the nursery, it is important to have good buoyancy and stay aware of where your fins are. Divers should be able to work with their hands while simultaneously maintaining good buoyancy. avoid touching the bottom when possible. Gloves are important to protect both the corals and you. There are lots of stinging creatures that like to hang out on nursery structures, so exposure protection is important. The final destination for all nursery corals is to go back onto the reef. We call this outplanting. Corals are outplanted on predetermined sites based on several factors. These include presence of herbivores or algae eaters, wave energy, and the health and diversity of the surrounding reef. This can be determined using in-water surveys, mapping data, and local knowledge. Outplanted corals should be free from tissue loss, discoloration, bleaching, or parasites. The first step in outplanting a coral is to find open space on the reef and clear as much algae from the spot as possible. Coral fragments can be outplanted using a variety of methods, including nails and zip ties, marine epoxy, and cement. The main goal is to stabilize each coral fragment so that it can attach to the reef. As the coral continues to grow, it will overgrow the epoxy or cement and attach itself to the reef. Using in-water surveys and photography, coral outplants are monitored initially and several months after outplanting to track survivorship, growth, and health impacts. Coral restoration is still a relatively new science, so it is important to continuously refine techniques as we scale up this process. During monitoring, it is important to note any unhealthy conditions of outplanted corals. These include predation, bleaching, or disease. Predation appears as distinct patches of white where tissue was recently eaten. On staghorn corals, fireworms will usually eat the tissue from the tips of the branches. On elkhorn coral, you can sometimes notice small, snail-shaped areas of dead tissue. Coral bleaching can make a coral appear bright white, but the coral still has all of its tissue. If you look closely, you will still see the polyps. Typically, once the stress subsides, corals will regain their algae and thus their color, but if the stress continues, the coral can starve to death. Coral diseases come in a variety of forms. Some, like black band disease or white band disease, present symptoms just as their name would suggest. Others can be harder to specifically identify. Stony coral tissue loss disease, first seen in the USVI in 2019, has caused widespread mortality in reefs in Florida and now in the Caribbean. This disease presents as lesions of tissue loss and affects many of the boulder and brain corals that are the backbone of our reefs. Research is underway to better understand this disease and to test possible treatments and interventions, including the use of antibiotics, removing lesions, and culling corals. The easiest way to help scientists and reef managers address disease and bleaching is to track both. If you are swimming, snorkeling, or diving, and you notice any unhealthy condition of corals on our reefs, report it to your coral reef managers. Here in the VI, you can go to vicoraldisease.org and submit a report. Detailed tracking of diseases can help scientists better understand them and can help dictate where to carry out interventions. Monitoring where bleaching does and does not occur each year can have indications for overall reef resilience and can influence what steps managers can take to best protect those reefs. Next time you're out in the water, tell us what you're seeing. Even reports of no disease or no bleaching are valuable information. Coral restoration projects around the world depend on the participation of the community to truly be successful. If you are a diver or snorkeler, you can help maintain nurseries, plant corals, and survey reef sites for signs of disease and bleaching. But you don't have to volunteer underwater to get involved. Land-based nursery facilities require a lot of maintenance with no scuba gear necessary. We also love getting volunteers involved in our education and outreach initiatives. And for those without a lot of free time on their hands, support for our coral reefs can come in everyday actions. Fight climate change by conserving fossil fuels, and support leaders who do the same. Reduce local impacts to reefs by keeping pollution away from our beaches, choosing sustainably caught local seafood, and being mindful of the reef when you are boating, snorkeling, or diving. It is our hope that these restoration efforts will maintain and enhance our coral reef ecosystems that are so vital to our islands and our people.